the last few days we've been looking at how to record the EEG, how to design an experiment, and especially yesterday we've been looking at how to compute event-related potentials. I've already explained how we look at the time course and how we look at the difference waves. And at the demonstration that I gave yesterday, I also showed topographies. And I think this idea of topographies is an important concept because it's the topographical distribution of the voltage over the scalp that provides information where the sources are in the brain. This is a figure that I've shown earlier. So if we have a source in the back of the head, that source is going to be stronger on the electrodes on the back of the head, and a source at the front of the head is going to be stronger at the electrodes at the front of the head. An electrode that's in between picks up a mixture, and the one at the back mainly sees the occipital activity, the one at the front mainly the high frequency activity. So we always have these mixtures between, if we're looking at the data at the channel level, so at the level of the electrodes or at the level of the scalp, we always see a mixture of underlying activity, and with source analysis, we're trying to understand where the activity is coming from in the brain. What I will do is in this lecture, I'm going to I'll first give a short motivation, an overview, or like background for why we're doing source modeling and how we're doing source modeling. And then there's two important components to the lecture. One is about forward modeling, and one is about inverse modeling. There's going to be a few equations in the lecture, but also without understanding the equations, you should be able to get the grasp what source modeling or source localization is about. Yesterday we looked at event-related potentials and we looked at ERP components, and they reveal the when and the where something is happening. So the brain is not a single unitary thing, it consists of many specialized modules. And by making smart experimental designs, we can basically elicit activity in specific regions in the brain. And by computing these different waves, for example, we can cancel out the activity in primary sensory areas, and we can try to look at activity in higher order cognitive areas, which are, for example, related to the brain function that we're studying, or which are related to the disease or the patients that we are studying. I've also shown that when we pre-process the data, we cut it into small segments, and then when we compute the event-related potential, we average over all those segments to get the ERP. When we look at one of the channels over all the time points, we basically can see when does specific brain activity start. So for example, if we have an N100 component, then around 100 milliseconds following stimulus presentation, there is a negative deflection, and from that we can conclude that around 100 milliseconds after stimulus presentation, the visual cortex starts processing the visual input. When we look at difference waves, then one channel can tell us when the activity becomes different in two conditions. So for example, the oddball experiment that we looked at earlier this week with the beeps and the boops, we can subtract the beeps and the boops, and we can look at when the brain gets surprised by the presence of the unexpected boop. And that would be around 150 milliseconds. The other dimension that we can look at is the topographic distribution. So rather than looking at one channel, we're going to look at all channels simultaneously. And this is where it becomes really important to have a lot of channels. The more channels you have, or the more electrodes you apply to the participant, the more accurate you can actually understand the topography and measure the topography, and also interpret where in the brain the activity is coming from. So we've been working with an 8-channel EEG amplifier, with a 32-channel EEG amplifier, and with a 64-channel EEG amplifier. And in general, I would say source modeling techniques, like I'm going to explain in this lecture, doesn't really make that much sense if you do this on an EEG system that has fewer than 32 electrodes. So from 32 electrodes upwards, I would say that these methods start working. But the more electrodes, the better source modeling techniques will work. This is an example of such a difference wave. I showed this yesterday, and this is a topography of the brain, sorry, topography of the scalp seen from the back. And the difference wave is for faces versus cars, and the activity around 170 milliseconds shows the processing in the brain that is specific for faces. And this activity can then be localized using source modeling techniques, or using fMRI, or using lesion studies to come from the fusiform face area. Localizing and understanding the ERP involves that if you find an ERP component, you want to characterize it in its physiological terms. So you want to relate the ERP to the brain. The time or the frequency of brain activity are directly related to the cortical activity. So the EEG is really a direct and instantaneous reflection of what the brain is doing. So for understanding the temporal aspects of an ERP, well, you just have to look at the ERP. But the location in the brain from where the ERP originates, that requires an interpretation of the scalp topography. And forward and inverse modeling help to interpret this scalp topography. So that's one important reason why we use forward and inverse modeling. The second important reason why we use forward and inverse modeling is that it helps to disentangle overlapping time series. And that's what I showed you before, is that if we have an electrode that is in between two brain regions, the electrode is going to pick up activity from both, and using source modeling we can disentangle this overlap. I've been talking about topographies, and yesterday at the end of the day we've been looking at some topographies. We've been looking at the topography of the auditory evoked potential, which is maximally over the vertex. We've been looking at the topography of the visually evoked potential, which is maximally over occipital areas. And here I'm just showing some topographies. So this is often how we look at them. Color-coded, going from blue to red with yellow and greenish in between, or color-coded from blue to red with white in between. What is an important thing to see here is that we not only have the color code, but we also have these isocontour lines. So these are lines where the voltage is constant, and these are also the lines that you would see on the topographic map of a landscape, where the lines basically reflect regions where the landscape is just at the same height. If these isocontour lines are very close to each other, we know that the landscape is very steep, so we know that we have to climb a hill or a mountain. And if they're very shallow, so if the lines are far apart, it is not so steep. So this is an important idea, because it's the spatial topography, which I'm going to show in the next slides, that relates to where the sources are coming from. So on Monday, Mikkel already explained equivalent current dipoles. And these equivalent current dipoles is how we think about small cortical patches that have synchronized activity. So the spatial topography that we might see consists of a positive deflection over the more frontal brain regions, a negative deflection over the more posterior brain regions, and then we can infer from this spatial topography that there's a dipole underneath. So by the way, this is the nose, so this is seen from the top, that there's a dipole which is pointing from the negative to the positive deflection. So what we can do just by very informally localizing the activity is that we determine the maximum of the negative and the maximum of the positive, and we can connect them, and then in between, that is where we can place the dipole, or that's where we can assume the dipole to be. So this gives opportunity for a few approaches. For example, that if the spatial topography shifts, well, that must mean that the dipole has also shifted. And it doesn't mean that the brain has shifted, but it means that the activity is now coming from a different brain area than I was showing over here. Or if the pattern rotates, it means that the equivalent current dipole also rotates. It doesn't mean that the brain rotates, the brain is just fixed, but it means that with this very complicated cortical folding pattern, that the dipole, which is in the cortex, the equivalent current dipole with which we model the cortex, is in a slightly different cortical fold that is oriented differently. Sometimes we see spatial topographies that look like this. So we see a spatial topography that is maximally or minimally at the vertex, and that if we compute an average reference, then the consequence is that it's like if it's most positive here, it is most negative around it. So in this case, the dipole is pointing towards the vertex, towards the top of the head. So it's what you can see here, the dipole is pointing towards us as we're looking at the screen. Another thing that we know, and that is what we can compute, is that if the spatial distribution is further apart, that corresponds to a dipole or an equivalent current dipole or a cortical patch of activity that is deeper in the brain. So by looking at these spatial topographies, we can already infer a little bit what the activity is. Of course, in reality, spatial topographies are not so clean as they are here. So that's why we need more sophisticated mathematical techniques to disentangle the spatial topographies and to explain them in terms of equivalent current dipoles. This is where biophysics comes. So this is a field that I've been studying a lot. So biophysical source modeling basically approaches this from that we have the body, the tissue of the body, or the tissue of the head in this case, and then that is seen as something that electrically can conduct currents. We have a source of the currents, so we have an electrical current that is generated by the brain, so that is the physiological source. And if this physiological source produces currents that flow through the body, then we can do an observation by placing electrodes on the outside of the body or outside of the head. We see this as a EEG potential, or in the case of MEG, we see this as a small magnetic field. From the perspective of the physics, if we go from the physiological source to the observation, we call that a forward model. Whereas if we go from the observed potential of fields, if we try to infer what the underlying sources are, that's what we call an inverse model. So we're using models to understand the relation between the sources in the brain and the EEG. The thing that you're probably mostly interested in is in the inverse modeling. So we do EEG recordings, and we try to interpret these EEG recordings by figuring out where in the brain the activity is coming from. But we cannot do inverse modeling without also doing the forward modeling. The forward modeling is a necessary ingredient for the inverse modeling. So I'm first going to explain the forward model. One of the ingredients in the forward model is the source model. And as Mikkel already explained on Monday, we have this cortex. In this cortex, we have neurons, and it's a specific class of these neurons, the pyramidal cells that have their dendritic trees very nicely aligned. So here you see histology of a very thin slice, and here you can see the individual neurons with the dendritic tree. And what happens is that if there's activity at the synapse, then we get a postsynaptic potential in the neuron, which is either flowing towards or flowing away from the soma. So this is the soma, the cell body. Each of these neurons, therefore, is a small battery. So you can just think of this as a very small battery with current flowing from the positive to the negative, or from the negative to the positive side. So these currents are so small that we cannot observe them, unless you do a patch clamp recording in an animal, or if you have a sharp tip electrode that you place directly in the brain of an animal. What we do with EEG, we look at this from a much larger distance. So rather than thinking about the individual neurons, we have to think about the larger scale. And this is where equivalent current dipole modeling comes in. So if we have a small, like a cortical column that consists of many neurons, what we can do is we can model each of these neurons as a dipole. And what we say is, well, since we're looking at it from such a large distance, we cannot really distinguish each of these individual neurons. So we're just going to model it with an equivalent current dipole, which is the summary or the summation of all of these individual neurons. And since we're measuring on the outside of the scalp, and the skull is in between, so the data is rather blurry, we can take this one step further. So rather than modeling an individual cortical column, we can also say we model an individual patch of cortex. And each cortical column in that patch is an equivalent current dipole, and that again we can summarize that with one equivalent current dipole modeling the whole cortical patch. So this works really well. The potential distribution that we can compute from such an equivalent current dipole is really very similar to the potential distribution from such a cortical patch. But at a certain point, if the patch gets too large, or if the patch starts getting curvature, of course the cortical sheet is highly curved, what can happen is that the center of mass, so the tail of the dipole, is going to be moved a little bit towards the center of mass of the patch. So it's not in the patch anymore, but it's just below the patch. So if you do source modeling using equivalent current dipoles, you should not be surprised that these equivalent current dipoles are not going to end up exactly in the cortex, because they're just an approximation of the activity in the cortex. They're not really modeling the cortex itself. The second element that we need in forward modeling is the volume conductor. And the volume conductor describes the electrical properties of the tissue. The reason why EEG works is because the head is electrically conductive. The conductivity of the different tissues in the head varies, but what we do is we make a geometrical model of the head. So we describe the model, we describe the shape of the head, and then we describe how the currents flow through this head. It is important here that the volume conductor model doesn't specify where the currents are originating from. So the source specifies where the currents are originating from. The volume conductor model is basically describing how the currents flow. And it also means that we're using exactly the same volume conduction model for EEG, where we know that the currents are generated in the brain, as for MEG, where we're also looking at the currents in the brain, but then through the magnetic fields. But we're also using the same volume conduction models for transcranial direct current stimulation or transcranial alternating current stimulation, or for transcranial magnetic field stimulation. So this is really about the passive conduction of the currents through the tissue. Very simple models describe the head as a sphere, and that is actually quite useful, but it's not so accurate, and especially it's not so accurate if we want to subsequently co-register the dipoles that we fit to the EEG to the underlying anatomy. So nowadays we're actually not using spheres so much anymore, but we're using more realistic geometries. Analytic computational methods for volume conduction problem allow for simple geometries, such as a sphere. So a sphere, we know exactly how to compute how the volume currents flow through it. And that makes us very accurate and very fast. We also have numerical computational methods for the volume conduction problem, which allow for realistic geometries. And those are the ones that almost everyone is using nowadays. And there's two methods, both have an abbreviation, that are widely used. And that's the BEM, or the boundary element method, and the finite element method, the FEM. And I'm going to explain these two methods just briefly. In the boundary element method, what we do is we say that the head is comprised of multiple compartments. And each compartment is assumed to be homogeneous, which means that the conductivity is assumed to be the same everywhere. And it's assumed to be isotropic, which means that the conductivity does not depend on the direction in which the current is flowing. Then the important tissues that we model are the skin, the skull, and the brain. And sometimes we might also include the CSF. But in general, we say, well, the CSF is just like part of the brain. Also here, I'm not making a distinction in the brain between gray matter and white matter, even though, well, white matter has like more fatty content, so the conductivity is slightly lower. White matter also has this fibrous structure, so the conductivity actually depends a little bit in which direction the currents are flowing. But this is something that the BEM is not very suited for. What we then do is that we make triangulated surfaces to describe these boundaries. And triangles are really convenient because with triangles I can make arbitrary shapes. So I can use triangles to make a cube or a sphere or a realistic shape such as a head. The strategy that we use for making these triangulated model descriptions is that we take an anatomical MRI, and for each of the voxels in the anatomical MRI we determine which tissue type it is. So we're going to color code each of the voxels, and we're going to split them into brain, skull, and scalp. And once we know these tissue types for each of the voxels, we can determine the boundaries, and we can describe the boundaries using the triangles. The computation of the BEM method depends a lot on the number of triangles. So if you want to have a very nice looking head, we use a lot of triangles. But if you want to have fast computations, we have to reduce the number of triangles to more reasonable numbers. Now that we have the geometry, we can assign the connectivities to the 3 different tissue types. So we say, well, the skull is more resistive, and the scalp and the brain are more conductive. And we can compute the volume conduction model. And this computation is independent of the source model. So this is something that you only have to do once. And then once you use it during forward modeling and inverse modeling, it is very fast. The second technique that I mentioned is the finite element method. And in the finite element method, we use small elements, and we would typically use tetraheders or hexaheders. So a hexaheder is basically just a cube, and a tetraheder is like a pyramid that has 4 corner points. So not 5 corner points like a regular pyramid, but it has 4 corner points. And the advantage of using these shapes is that we can make arbitrary complex geometry. So this is a head that is built from Lego bricks. And what you can imagine is that these hexaheders and tetraheders, that we use them as if they were Lego bricks. So on the left, you see a tetrahedral model. And a nice thing of these tetraheders, similar as these triangles that I showed before, is that they can describe very complex geometries, while at the same time also modeling this curvature. With hexaheders, this is a little bit more difficult, because hexaheders by themselves are cubic. So with hexaheders, a lot of the models actually don't model the curvature, but they basically result in a more brick-like appearance, as you see here. But there are also FEM methods for shape-adjusted hexaheders, where you can also model smooth surfaces. Tetrahedral models are in general the most accurate because of the better approximation of the poorly conductive tissues such as the skull. With the finite element method, each of the elements that we use can have its own connectivity. And that makes the finite element method the most accurate numerical method, but its computation is also quite expensive. There's a lot of elements that we need to describe the head, and that means that you need a computer with a lot of memory, and you just have long computations that you have to wait for. Also, the geometrical processing of the anatomical MRI is not as simple as with the boundary element method. So I would say that in most cases, the boundary element method is good enough. But if you have very specific cases, for example, if you have patients with a brain lesion, where part of the tissue is lesioned and is replaced by blood or by CSF, then that is something that you have to take into account. And in that case, you would have to use a finite element model, because the boundary element model does not allow for modeling it at such a level of detail. I've already said that, so we're looking at volume conduction of the EEG. And earlier this week, we've explained that the EEG, with EEG, we're measuring the potential difference between electrodes that we place on the scalp. And these potential differences, they basically correspond, or they relate to the currents that are flowing through the skin or through the scalp. However, the skull is very poorly conductive. So there's only a small fraction of the current that is generated by the brain, which is flowing through the skull into the scalp. And that's also one of the reasons that EEG signals are so small. And if we consider this during modeling, we have to model the skull and the skin, or the skull and the scalp, as accurately as possible. So even though we're interested in the brain, it's the skull and the scalp that we need to model, because they really affect how the potential distribution is when we record it with the electrodes. Throughout the week, we've also, for a few times, been mentioning magnetoencephalography, or MEG. MEG is a technique, so this is a technique that I'm mainly working with, where we measure not the electric fields by placing electrodes on the scalp, but we measure the magnetic fields around the head with a large scanning device like this. MEG is a very expensive technique. In the Netherlands, we only have 2 MEG systems. In Denmark, there's only 1 MEG system. So MEG is really quite a a unique opportunity, but also expensive, and it's only being used by very large imaging centers. But the advantage that we have with MEG is that the magnetic fields are not really disturbed by the volume conductor. So MEG produces data which is much more focal. So it is much easier to distinguish between two different sources if these sources shift a little bit. And furthermore, with MEG we still have the volume currents. The magnetic field itself is not distorted, but with MEG we basically have the same characteristics of the volume currents. So only a tiny fraction of the current passes through the skull. And therefore, when we make an MEG model, we typically ignore the skull and the skin because they're not really relevant, because there's just such a small amount of current flowing through the skull into the skin. And that means that with MEG we have a much more simple volume conduction model, because we only have to model the inside of the skull. So the whole source modeling with MEG is much simpler than it is with EEG. It is, however, important that with MEG we still have to consider the magnetic fields that are coming from the equivalent current dipoles, but also from the volume currents. And there are some disadvantages in MEG to that as well. For example, that MEG cannot really detect activity in very deep brain regions, whereas EEG has no difficulty in detecting activity from deep brain regions. Are there any questions so far about the forward modeling or about the source model that we're using? Because if not, then I want to move on to the inverse modeling, which is more, again, like more related to the biophysical models, but it's trying to relate the recordings that we've done to the activity in the brain. So what we're now doing is we're going to start from our observations, from our event-related potentials, and we're trying to infer where in the brain the event-related potentials were generated. What I want to start off with is a small demonstration. This is a demonstration that I recorded when I was a PhD student. So this is a demonstration actually showing how source localization works for a very wide audience. We just had a public day, and during this public day we wanted to show how source localization was. And what we did is we got a fish. So this is an elephant fish, and I've been told that they naturally live in the Nile. Actually, it's not something that I can confirm. In the Netherlands, we just have them in the animal shop. And this fish is special because it has a side organ with which it senses its environment. So a little bit like how bats use ultrasound echo navigation, this fish uses electric pulses to sense its environment and to sense prey. And I'm just going to keep my microphone closer to the computer. So what you hear is this clicking sound, and each of these clicks, I'm just gonna turn it off again. Each of these clicks is the fish emitting a small electric pulse. And what we did is we put the fish in a fish tank, and we connected the fish, we drilled holes in the fish tank, and we connected it, so this is 25 years ago, to an old EEG amplifier. So this EEG amplifier was already old 25 years ago. So this is an analog EEG amplifier, and what it does is it records the potential distribution around the fish tank, and that is something that we can also model. So what we did is we made a demonstration where we're trying to localize the fish on basis of the potential distribution around the fish tank. So here you see the computer on which I did my PhD research, and on the screen, so this is the EEG system, here you see the fish, and on the screen you're seeing a real-time visualization of dipole fitting. So in one corner of the fish tank there's a heating element, and the heating element is also used here to indicate where the fish is. So every time that the fish emits an electric pulse, we would record the spatial topography, and we would fit a dipole to the spatial topography. So the dipole not only shows where the fish is in the aquarium or in the fish tank, but the dipole also shows in which direction the fish is pointing, because the dipole of the fish, or the equivalent current dipole that we model to the fish, is pointing along the length of the fish. So here you see, for every pulse, you see that the position is being estimated, that the strength is being estimated, and you also see the relative difference, which indicates how well the dipole explains the potential distribution that we've recorded. So this is not really event-related potentials, because the fish is not showing like a continuous potential distribution. This is much more like epileptiform discharges, like very short spiking activity. But also there, for each spike, you would want to localize where in the brain is the spike coming from, because that might tell you where in the brain the lesion is. So in this case, it is very transparent, like literally transparent where the fish is, and we can confirm that the direction that the fish is swimming, so you can see it here, and the location of the fish in the fish tank, that it corresponds with where the fish is. The problem that we have to solve with activity in the brain is that we cannot see where the activity is coming from. And that's where we use these mathematical techniques like inverse modeling. There's a number of inverse methods that we're using. And the ones that were initially introduced and that are nowadays still used, especially for focal epilepsy, are single and multiple dipole models. And the idea with single and multiple dipole models is that we take these equivalent current dipoles, we compute the potential distribution that we expect, and then we change the parameters such that the error between the model and the measured potential is as small as possible. And I will explain these in more detail. Then we also have another family of methods, and those are distributed source models. So rather than assuming that the activity comes from a single point, with the distributed source model, we expect the activity to be distributed throughout the whole brain. The problem that we have with that is that we have a perfect fit of the model to the measured potential, and that means that we need additional constraints such as the source smoothness or the amplitude or the power. A third family of source reconstruction methods is spatial filtering. And with spatial filtering, we're also considering that there can be activity everywhere in the brain, but rather than estimating it all at the same time, what we're doing is we're scanning the brain. So for every location in the brain, we're putting the dipole there, and we compute a spatial filter. And for every location, we try to estimate how much activity is coming from that region, while simultaneously suppressing the activity from other regions. And these beamforming methods, they come in a variety of classes, such as linear constrained minimum variance beamforming, or dynamic imaging of coherent sources, or multiple signal classification. And each of them has its own characteristics and its own assumptions. What I primarily want to focus on in this lecture is on single and multiple dipole fitting, because those techniques are relatively robust and work well for data that doesn't have too many channels, which I think is likely going to be the data that you're going to record. Techniques such as beamforming require a lot of channels and are usually done with MEG systems rather than with EEG systems. So beamforming doesn't really work that well for EEG data. Distributed source modeling is a little bit in-betweenish. I will touch upon beamforming and distributed source models towards the end of the lecture, but I'm primarily going to focus on how dipole fitting works. And to give you an impression, so this is how results of dipole fitting look like. So we might have a spatial topography with a strong negative deflection at the back and a large deflection at the front. And then using dipole fitting, we can, for example, so I suspect that this is alpha activity, we can fit two dipoles, which then end up in occipital cortex. And if we have dipoles, so this is another example, we can plot where these dipoles are relative to the brain, or we can plot where these dipoles are relative to the anatomical MRI. So here it's shown relative to a template anatomical MRI, and here it is shown relative to an individual participant's MRI. So ideally, we would want to know in each of the individual's brains where these dipoles are, but in practice, I would expect that you're not going to get anatomical MRIs of each of your EEG participants. So for you, it's going to be more similar to this, where you're going to use a template anatomical MRI and a template model and also template electrode positions. So the accuracy is going to be less, but it's still going to be helpful to interpret the spatial location of where the activity is coming from. Dipole modeling is, in a way, it's not very different than parameter estimation. So if I show you this, and if I ask you, can you predict y given the values that we have observed? Then you would say, well, yes, I can fit a straight line, I can do regression. And using regression, what I would do is I would fit a number of parameters. I would fit the intercept, I would fit the slope. And with this intercept and the slope, I'm making a prediction of y. And the intercept and slope are chosen such that the error that we have for the model is the smallest compared to the data. So that's what parameter estimation is about. And that's actually also what dipole fitting is about. In dipole fitting, we don't have a slope and an intercept, but we have a source model. And the source model has a number of parameters. So one of the parameters is the position of the dipole. Another parameter is the orientation of the dipole, because the dipole is creating current. And if the dipole is in another direction, the current is flowing in another direction. And then the third parameter is the strength, like the amount of current that is generated by the dipole. What we can do using the forward solution is we can compute the model data, and then subsequently we minimize the difference between the actual data and the model data. And that's going to give us the parameters of the source which we can interpret. So, as a toy example, it's just like fitting the line, and it's going to give us A and B. But now rather than having A and B, we have the position of the dipole, the orientation of the dipole, and the strength of the dipole. Just a little bit of math here. So I've told a few times that the EEG signal is a linear superposition. And this linear superposition, we can think of this as a combination of multiple sources. Here I'm showing the equation that we always use. So the data y is, for example, 64 channels times 500 time points. And the data is modeled as a spatial topography of one source, and that's the spatial topography of the model is called the lead field matrix. And if we don't know the orientation of that source, we're going to have a lead field matrix that has 3 columns. One for the x direction, one for the y direction, and one for the z direction. And the lead field matrix is multiplied with the source strength. And the source strength of each of the sources is then 3 for the x, the y, and the z direction times 500 time points. So that means that this is the contribution of one source. This is the contribution of a second source, etc. So we have multiple sources that are contributing, and then we also assume that there's going to be some noise. So the noise here is again a matrix, and then the size of that matrix is the same as the size of the data. The lead field, that's what we compute using the forward model, and the lead field depends on the position of the source. And then what we do is we compare the model data y with the measured data. Okay, so we have multiple parameters. And what I'm going to do here is I'm just going to say, well, these parameters, I'm going to call them the letter zeta. The zeta is the collection of all the parameters. So it's the position and the orientation and it's the strength. And if we just like plot the parameters along the horizontal axis, of course there's multiple parameters, but that's difficult to plot. So I'm just like plotting all the parameters along the horizontal axis. Then what you can imagine is that we compute the model potential and we compare that with the measured potential. For each of the electrodes, we take the square and we sum it over all of the electrodes. So we take the sum of the squared error. That's the same what we do with regression. And then what we want to do is we want to minimize the sum squared error. And that's going to give us the parameters at which the model is best capable of explaining the data. The problem, however, that we have is that this error landscape, we cannot compute it everywhere. And there might be some location where it's minimal and other location where it appears minimal, but it's not really minimal. The first step that we then perform is a grid search. So we can at many discrete locations, what we can do is we can estimate what the lead field is, and therefore we can estimate how well the model for a dipole at that location is able to explain the data. So in the first step, we do a grid search. And if you imagine, if you, for example, go from medial to lateral, and if you take 100 possible locations, I'm just going along this axis, like from left to right, now I would have to compute 100 different lead fields. But if I also look at inferior superior, or if I also look like in 3 dimensions, I have 100 times 100 times 100 different locations in the brain at which I have to perform this computation. And that's 1 million locations. Imagine that each of these locations imagine that each of these computations takes about a millisecond. Well, 1 million milliseconds is 1,000 seconds, and 1,000 seconds is about 20 minutes. Well, 20 minutes is doable. We can wait for 20 minutes. It becomes problematic if we have two dipoles, because then the first dipole can be at 1 million locations, and the second dipole can also be at 1 million locations. So all of a sudden, you have 10 to the power of 12 locations, or 10 to the power of 12 possible source configurations that we have to consider. And then, of course, we would have like an infinite amount of time. So that means that for multiple dipoles, the grid search is not an efficient strategy. It just computationally takes too long. And that is why we're using another approach. So we don't really know what the field distribution is, what the error distribution is, because we cannot compute it everywhere. But what we try to do, we try to find a starting point. And at this starting point, we're going to compute around the starting point in which direction the error is going to go down. And this is called gradient descent optimization. It's like walking in the mountains and trying to find your way back to the valley. Well, you don't really know where the valley is. Perhaps it's very foggy, perhaps you cannot see it very well. But what you're going to do is you're just going to walk downhill. And just keep on walking downhill until you reach the lowest point. And this is the lowest point, and that's the point that we're trying to find. Something that you might see is that there's not really a guarantee that it's going to be one lowest point. There might be multiple lowest points. And if you're going to start here, you might end up here. And this is not the optimal location. So we need additional strategies to help us to find the optimal location. So one of these additional strategies is that we often start with a grid search, or that we start with an initial guess, like knowing possibly results from literature, and we're going to place the dipole where we more or less expect it to be, and then we're going to start walking from that direction, walking from that point into the direction where it's going down. So with a single dipole, we can scan the whole brane. Actually, it doesn't take 20 minutes. It's faster than that. It takes about perhaps like 3 minutes or 5 minutes. So we scan the whole brane relatively coarsely, for example, with 1 centimeter steps. And then we do this iterative optimization with the gradient descent method. If we have two dipoles, then depending on the task that we're doing, but if it's sensory activity, we very often can assume that there's symmetric activity in the brain, so that the homologous areas in both brain, in both hemispheres are active, so we can make a symmetric dipole pair. And if we make a symmetric dipole pair, if I move one dipole, the other dipole automatically moves along with it. So that means that I can scan the whole brane with a symmetric pair, and actually I only have to scan one hemisphere because the other hemisphere automatically goes along. And again, we can use that as a starting point for the iterative optimization. If the data needs more dipoles to be explained properly, then we often use sequential dipole fitting. And sequential dipole fitting basically assumes that we describe every cortical patch with an equivalent current dipole. So these are the individual dipoles, and this is the equivalent current dipole. And what we do is we look at the ERP, and we realize that the ERP is basically evolving over time. So what you see here is two cortical patches. The red one is activated slightly earlier than the blue one. So at the scalp level, these two time courses, they would overlap. And we can look at the topographies, and if you look at the topographies, and that is something that you might have seen yesterday when I showed the movie of the ERP, is that you have like a rotating pattern. So the pattern from here to here, so the nose is, by the way, the nose is pointing here towards the right. But the pattern starts in a particular way, but then it rotates over the scalp, and it basically becomes another spatial topography. The strategy that we use in sequential dipole fitting is that we're going to first look at a very early time point. So we're going to look at the activity very early in time, and we're going to try to fit that with a dipole. So this is the potential distribution that we can compute. So we scan the whole brane, we find this location as the best, we make the forward model, and using the forward model we can compute the potential distribution. Using the inverse model we can estimate the source waveform, and we can compute a difference wave. So we can compute Like, how well does the model explain the data? And what you can see is that the model explains the data really well in this early window, but a little bit later, the model starts failing. The model is not really able to explain the data that well. And if the model starts failing, well, then we know that we need a second dipole. So what we then do is we keep the first dipole at the location where we fitted it, and we add a second dipole. Again, we compute the forward model for both dipoles now. So that basically means that we can use a summation of these two scalp topographies to explain the data. And that's going to give us the source waveforms. And with these source waveforms, the residual is 0. So we're able to explain the data perfectly well. So the number of dipoles that you need to explain the data, that is always a bit of a question. It really depends on the quality of the data. If you have good quality data, you can fit more dipoles. You can see more detail, and you can also fit these dipoles more accurately. But estimating the number of dipoles that is necessary to explain the scalp topography, that is still quite a tricky business. But this approach allows you to study the spread of activity through the cortex. So sequential dipole fitting is used to explain spread of activity in a cortex, where we assume that the activity starts small in a relatively focal area, for example, in the sensory area. So we explain the earliest ERP components with a single equivalent current dipole. And then we assume later activity to become more widespread. There's more brain regions going to be involved. And then we add equivalent current dipoles to explain the later ERP components as well. And then if we add additional equivalent current dipoles, then we estimate the positions of these new dipoles as we're adding them. And then every time we re-estimate the activity of all the dipoles. This approach is iterative. So we basically have to add a dipole and then start again, and then add a dipole and then start fitting again. And it's interactive. And that makes it also subjective. This subjective nature of like when do you add new dipoles, how many dipoles to add, means that this is a technique for which you require some experience. And it's also a technique that some people might not like because it depends on the choices that you make as a researcher. But still, this is a technique that is very valuable, especially if you're using EEG data with a relatively small number of channels. Because with a relatively small number of channels, we just need a simple source model, and dipole models are the simplest source models. I also mentioned that we have like a totally different approach, which is distributed source models and beamforming. And in distributed source models and in beamforming, we're making a different assumption. So with dipole fitting, we assume a single or a few point-like sources, equivalent current dipoles. But what if we assume that there can be activity distributed over the whole brain? So we know that the brain consists of the white matter and the gray matter, the cortex, and that the activity of EEG is generated in the cortex. So we can also take the starting point. Let's assume that the whole cortex is active. And then rather than assuming that we have to localize where the cortex is, we can just say, well, we already know where the cortex is. The only thing that we have to do is estimate the activity in the cortex. So estimate the strength of the dipoles. And that results in images like this. So what you see here is a distributed picture of activity. This is from one of the field trip tutorials. And it can be shown on an individual anatomical MRI, like it's done here. So this is activity in the language task. But you can also show this activity on a rendered cortical surface. So using anatomical MRI processing techniques such as FreeSurfer, you can extract the cortical surface and you can project the color-coded values, you can project them onto the cortical surface, and you can do more fancy analysis such as inflating the cortical surface so that you can look well into the sulci as well. With distributed source models and beamforming, the position of the source is not estimated as such. We assume a predefined grid. So either we place dipoles everywhere in the brain and we say, well, so this is where the activity is going to be, or we place the dipoles on a cortical sheet if we have it, and then we say, well, we know that the activity must come from the cortical sheet, so that's where the dipole should be. And then we estimate the strength at every location. In principle, this is mathematically relatively easy to solve. But the problem here is that we have many more unknowns, many more parameters that we have to estimate than knowns, than the number of measurements. And as a consequence, we have an overdetermined problem, and there's an infinite number of solutions that can explain the data perfectly. And that means that we need additional constraints or assumptions. This is, for example, one of these distributed source models. So what you see here is you see the cortical sheet, and the cortical sheet is also extracted from the anatomical MRI, but the cortical sheet here is not used as the volume conductor, but it's used as a source model. And what we've done is that every corner point of the triangles that describe the cortical sheet, we've placed a small dipole. So you see all these small dipoles here. And the task that we then have is we have to estimate the activity at each of these dipoles. Let's again look at the mathematics. So we have the data, which we can model. The data is, for example, 64 by 500. But now we have many more dipoles than we had before. Each of these dipoles has a lead field matrix. So each of these lead fields, so the total lead field matrix then becomes very large. So there's many columns in this lead field matrix because we have many dipoles. And that also means that with such a large lead field matrix, as I'm using here, we have to estimate the strength. So the lead field matrix can be computed, but the strength needs to be estimated. And what we have here is that we have 8,000 times 500 parameters that need to be estimated. But the starting point is only 64 times 500. So we have many more parameters than we have data points. And this is an overdetermined linear system, and that's why this is challenging. It is a linear estimation problem. So we have this distributed source model with many dipoles throughout the whole brain. We can estimate the strength of all the dipoles, but the problem is that since we have an infinite number of solutions, the data and the noise can be perfectly explained. And again, thinking of this as a regression problem, earlier I showed you linear regression with only a slope and an intercept. But here I'm just extending this and I'm fitting a polynomial. And the more terms I add to this polynomial, the more wavy this line can become, but also the better the line can explain all the data points. That doesn't necessarily mean that the green line is a good model for the data, because what's going to happen is it's also going to model the noise. So modeling the noise is really a problem, and what you also can imagine is like the more terms I add, I can even make many more solutions with very large fluctuations that are still going to fit all the data points perfectly. So that's the situation that we have with an overdetermined model. So to find a unique solution, to find one solution rather than to find an infinite number of solutions, we have to make additional assumptions. And the general assumption that we make is that we want to have the simplest solution, so the solution with the minimal overall norm. And the norm is a mathematical concept which expresses the overall amplitude of the power of all the sources distributed in the brain. So what we do is we compute the estimate that has the minimal norm. So we're trying to estimate the activity under the constraints that the overall activity in the brain is as small as possible. So we're basically making the assumption that the brain is efficient and that the brain is not using excess activity out everywhere. And if we want, we can use additional assumptions, for example, about the noise, so that the solution should not explain a fraction of the data because that fraction is noise. And we can also make assumptions about the distribution of the activity. So if we have a priori knowledge, we can weigh the distribution and we can compute a weighted minimum norm solution. So with distributed source modeling, the source is really distributed throughout the whole brain, and the method that we use to estimate it is by estimating the activity at all the locations simultaneously. With beamforming, we're also assuming that there can be activity everywhere in the brain, but rather than computing the activity at all locations simultaneously, we compute it for each location separately. So the approach with beamforming is that we scan the whole brain, and for every location where we consider a dipole to be possible, we compute what is called a spatial filter. So the day before yesterday, during the pre-processing, I talked about filters. I talked about how we use a low-pass filter or a high-pass filter. You can also think about a spatial filter. Like a spatial filter zooms in on activity coming from a specific location in space while suppressing activity coming from other regions in space. And using the spatial filtering approach, we still need additional assumptions. And for spatial filtering to work well, we typically make the assumption that the sources are uncorrelated or are maximally independent. And that is not always the case in reality. So beamforming as a technique works well, but beamforming as a technique might not match the data. And especially in event-latent potentials, where we have multiple brain regions operating in parallel, the assumption of independence is often not met. So beamforming is not a technique that works very well for ERPs. Okay, so I've explained 3 different source localization methods: dipole fitting with 1 or a few dipoles. I've explained distributed source estimation using a minimum norm estimate. And I've explained distributed source estimation using beamforming. Okay, so that's of course ideal. That's not what you want to hear, that there's different methods. Each of them has different assumptions on the actual sources. What you want to know is which method should I use? Because you don't want to use 3 methods, because these 3 methods are going to give 3 different answers. The challenge here is that these methods, they make assumptions, and your data is different from other people's data. So the method that is best for you might also be different from the method that is best for other people. So that means that you have to know something about the data, you have to know something about the task in order to select the optimal method. So what I had in mind is just to shortly discuss some of the practical guidelines for selecting a method. So perhaps you can go in small breakout groups, just organize yourself like in groups of 4 or 5 or so, and then think about the experimental task that you've been like discussing earlier this week, and which method do you think that would help to localize the activity in that experimental task, to figure out where in the brain that activity is happening. So let's just take 5 or 10 minutes to discuss in your own experimental ideas which of the source reconstruction methods you would prefer to use and how you would approach this. So just returning to where we started off from. So yesterday I explained event-related potentials, and if you find an ERP component, you want to characterize it in its physiological terms, you want to relate it to the activity that you have in the brain. And time is the natural characteristic of the event-related potential. You directly see the amplitude of the ERP versus time, but it's this location that requires interpretation of the scalp topography. The lecture is called source localization, but actually there's more than just localization. There's other questions to address as well. So forward and interverse modeling help to interpret the spatial distribution in terms of the location that generates these spatial distributions. But they also help to disentangle overlapping source time series. So if you imagine that you have a distributed network of brain regions, of specialized brain regions, then using the ERP method combined with source reconstruction, you can try to get closer to assigning to each of the regions that you see here, the activity that is related to that region, so the time course of activity. And of course, then if you're able to design an experiment, for example, comparing patients to healthy controls, you can better study where in the brain specifically a patient, like a disease-specific deficit is related to which cognitive function. So that's how we disentangle the activity. So it's not only about localizing, but it's also about assigning the activity to the correct location in the brain. And that's what I just mentioned with these virtual channels. These virtual channels are a very efficient strategy of assigning the activity to a certain location in the brain. Are there any questions about this?